Simposio número 5, que es de señalización de calcio, impacto en la fisiopatología de células y tejidos. Quiero agradecer también la colaboración de mi coequiper Luis Bonano, del Centro de Investigaciones Cardiovasculares de La Plata. Mi nombre es Darío Krampf y soy del IBR de Rosario. El simposio que estamos llevando adelante va a constar de 4 oradores. El primero de ellos es William Lauch, del Institute for Experimental Medical Research, University of Oslo, Norway. It's a pleasure to have you here with us, Bill. So Bill is going to give the talk in English. You're welcome to write in the chat the questions either in English or in Spanish. I will translate it to English. So the questions going to be asked at the end of the talk of each speaker. William, of a project named Controlling Cardiomyocyte Dyadic Structure, which in Spanish is Control de la Estructura Diádica, supongo que se traduce de los cardiomiocitos. Bill, it's your time now. It's a pleasure to have you with us.
Thanks very much. It's great to be here, and I'm sorry that we can't meet in person right now. It would have been wonderful to in Argentina. As mentioned, I'm joining you from Oslo, Norway. You can see it's already well into the evening here. We're getting into a dark time of year. In my group, we work in cardiomyocytes, which are cardiac muscle cells, and we are really interested in their structure and how it controls calcium homeostasis, how it works in the healthy situation, and how it's changed in disease. So, to understand how calcium moves around, I have this simplified diagram. This is how contraction is triggered in the cardiomyocyte. You have the surface membrane shown here, and it has these tubes which extend down into the cell called T-tubules. You can see them in the previous diagram I showed you. And they contain a number of important proteins that move calcium around. You can also see here the sarcoplasmic reticulum where calcium is stored. So during each heartbeat then, the electrical activation moves down into the T-tubules and it causes these L-type calcium channels to open, and that brings calcium into the cell. This calcium then binds to the ryanodine receptor, calcium to open, and a large amount of calcium is then released from the myofilament from the SR, binding to the myofilaments, triggering their contraction of the whole cell, contraction of the whole heart. So all the action happens right in here. It's called the dyad, where these two membranes, the T-tubule and the sarcoplasmic reticulum, the SR, are very, very close together, only 12 nanometers apart. And this makes for a real challenge when we want to understand where these different proteins are located. So as an example, we've used confocal microscopy for many years, starting back around 1990 when people started to get these microscopes available to them. If we use a confocal microscope and we stain for these different proteins, here you can see the L-type calcium channel and the ryanodine receptor and the sodium calcium exchange. These two sit in the T-tubule. This one, of course, sits in the SR. Now, when we look with confocal, it looks like the proteins are colocalized. It looks like they're on top of each other. Now, that can't be right. They're in different membranes. So why would we get that impression? Well, it's because the resolution of the technique is not very good. It's about 250 nanometers. And as I told you, those two membranes are only 12 nanometers apart. So if we really want to understand where everything is, we have to use techniques that have better resolution. And that brings me to super resolution imaging. Now, to understand this point, we should know that whenever you look at a point of light with a microscope, it diffracts in a very stereotypical manner. So we call it the point spread function. So if this is your light source, it's for example, an antibody with fluorophore attached to it, bound to your protein of interest. That point of light spreads out according to the point spread function. So it means that if you have 2 points of light that are very close together, their point spread functions overlap and you can't tell them apart. So the super resolution imaging technique we use is called D-storm. And with that, you get the fluorophores blinking randomly. And because they're all randomly blinking, they don't overlap with their neighbors, and you can very accurately pinpoint the center of each blink. So now you get a resolution of around 25 to 30 nanometers. It's around 10 times better than you get with a confocal microscope. So here's an example. If you look at the calcium release channel, the ryanodine receptor, with turf imaging or with confocal, we know that these proteins are present together in clusters. But when we look with these low resolution techniques, all we can see is a single blurred image. We can't discern the individual proteins. Now, if we do D-storm imaging on the ryanodine receptors, we collect all of these blinks randomly, maybe over 15 minutes or so. And as I said, we pinpoint each of these blinks and then we reconstruct an image. Here you can see the same part of the same cell. You see how much better the resolution is. Now we can see where the individual proteins are located. So it's a very powerful technique. There's another trick we can use, and that is we can also do this in 3 dimensions. And you insert a prism into the light path, and that divides the point spread function into 2 lobes. And depending on the depth in the sample, these 2 lobes rotate around each other, and that gives you information about the Z position. And the computer then can make a reconstruction in the Z axis. So here you can see, if we look again at the ryanodine receptors in 3D, you see these clusters of proteins along the Z lines and cardiomyocytes, and also at the cell surface. And they have really interesting and variable shapes. So if we look at them together with the teachables, remembering that there's a very close, tight interaction, we can image that in 3 dimensions across the cell. And that's what we're showing here. This is a reconstruction based on our D-storm imaging. The teachables are shown in blue. The ryanodine receptors are shown in orange. You see the SR outline the ryanodine receptors at the dyads. You see that things are a lot messier than we try to make them look in those simplified diagrams. This is from my postdoc, Xin Shen's work, which was published 2 years ago. So it's an interesting way to get more information about where things are located. And I hope I've convinced you that there's a very precise and nanoscale arrangement of these different proteins that control calcium. It's very important for regulating contraction. So I wanted to just mention in the next few minutes how this changes during disease. And we've looked especially in heart failure. And these days we understand that there's actually 2 different types of heart failure. There's the classic heart failure, which we now call heart failure with reduced ejection fraction. And that's the condition where the contraction of the heart is weaker, so it's less able to eject blood. You have a less powerful heartbeat. That's different than the other type of heart failure, which about half of patients have. We refer to it as heart failure with preserved ejection fraction, HFpEF. And HFpEF, the contraction of the heart is quite good. Problem diastolic dysfunction. So we'll talk about what happens to the cells and calcium handling in these 2 different conditions. So first, a slide from some of my early work during my boyhood when I first moved to Norway, and we were interested in what happened to the teachable structure. So we had healthy mice, and we had those mice with heart failure with reduced ejection fraction, HFrEF. And what we saw is that the teachables in the healthy situation are very, very well organized, but in HFrEF, there's really marked reorganization. You see that they're disorganized. There are these weird gaps between them, and lots of teachables. So that's interesting. We saw this in mice. We've since seen it in rats. It's been shown, I think, in about 25 different studies of animal models. But importantly, we can now also see it in patients. And this is in our recent work published earlier this year, where we've looked at teachables in healthy patients and patients undergoing heart transplant for HFrEF. You can see again, like in the animals, they have fewer teachables and they're often in the wrong orientation. So what then does this mean for calcium homeostasis? You remember this diagram I showed you before? What I showed you was just part of it, 1 teachable next to the junctional sarcoplasmic reticulum. So of course, in the healthy situation, these teachables and these dyads are located at regular intervals along the length of the cell. And at each of these points, the action potential arrives, triggers the opening of calcium channels, calcium influx, and calcium release very evenly along the length of the cell. So in HFrEF then, you lose teachables. Here's a teachable that's missing. We know that the ryanodine receptors are still present there. So what happens is that you get triggering of calcium release first at these sites where the teachables are present, and you have to wait for the calcium to diffuse into the gaps where you then get delayed calcium release. So you get a desynchronization of the calcium transient. It gives you a slower calcium transient. It gives you a slower contraction and a weaker contraction and a weakened power of the heartbeat. So we think it's a really important mechanism for HFrEF. Now, I talked about these higher resolution imaging techniques that we can now use, such as super resolution imaging. And I showed you how we can image individual calcium release channels, the ryanodine receptors. And in the normal heart, the healthy heart, these are grouped together in clusters. We know that this allows them to open and close together to make what we call calcium sparks. Now, what we saw is that in HFrEF, there's a marked disruption of these structures. So instead of having very solid large clusters, you see that the clusters disperse. We call it a dispersion of RYRs. You get many small clusters, and they're nearby each other. And this gives you less efficient calcium release. Let me show you very simply with these two videos here what it means. So if you look at the left side, this is when the calcium channels are close together, and they tend to open and close together, while those on the right side, they give you sort of a wave-like calcium release as calcium spreads gradually between them. So again, if we go back to the whole cell, we can see these effects if we look at T-tubules and calcium at the same time. So here we have, stay in the T-tubules, they're shown white, and we have the calcium over top. And here you can see in the healthy heart, the T-tubules are really good at triggering calcium release. Lots of calcium release from all the T-tubules. And in HFrEF, you have the problem that the T-tubules are in the wrong orientation, but also we see they're not very good at triggering calcium release. Some of them release calcium, but many of the others, the calcium we're seeing there is just diffusing in from out of plane. So we think that this is linked to these problems with the ryanodine receptors. The spreading apart, the dispersion of the RYR cluster gives you much less efficient triggering of calcium release. So just quickly, I'd like to say what we think causes some of these changes in HFrEF. Now, it's been widely known that there's a high workload placed on the heart during HFrEF. And we measured this looking at the wall stress. And in the normal heart, we looked with MRI and blood pressure measurements, and we could estimate the wall stress across the left ventricle. We saw that it was very low in the normal heart, but in HFrEF, we could see that it was higher. Here's where there's an infarction, this very thin region here. So the ventricle dilates, that increases the wall stress, but it's especially high here close to the infarct where the wall tapers down towards the very thin scar. And we could see, if we look here, what we call the proximal zone next to the infarct, it's here that the T-tubules were most markedly disrupted, and also the calcium release was most desynchronous. And as you move further away from the infarct in towards the the distal zone, where the wall stress is lower, here the T-tubule density was better maintained, as was calcium release. So this is only a correlation. It doesn't prove that wall stress is important. So to look more into this, my PhD student, Marianne Ruud, she studied papillary muscles exposed to different amounts of stretch. So when she stretched them a lot, exposing them to high wall stress, like you would see next to the infarct in HFrEF, it caused the T-tubules to be degraded. You got a lower T-tubule density over a couple days of culture. When she stretched them just a little bit to reproduce the normal heart situation, the T-tubules were maintained. So this suggests to us that the high workload, the dilation, the thinning of the wall that happens in HFrEF signals the destruction of the structure of the cardiomyocytes. It's a problem. So I said then that we would compare the HFrEF and the HFpEF situation. So you remember HFrEF, heart failure with reduced ejection fraction. There's a weakened contraction of the heart, and you have a dilation of the ventricles and a thin wall. The situation is very different in HFpEF. Here you see there's a hypertrophy, the walls are thicker, the chambers are not dilated. So there's no elevation of the stress on the wall. So you might think then the situation with the T-tubules should be very different, and that's in fact what we've seen. This is from Miko Frisk's article published earlier this year. We looked at patients, control patients, and those with HFpEF who were undergoing open heart surgery. We stained the T-tubules. Lots of T-tubules present in the healthy individuals, of course, but actually more T-tubules in the HFpEF patients, completely opposite to the situation in HFrEF. supporting that the high wall stress that happens in HFrEF is an important signal that causes the T-tubule loss. Now, remember I told you though that in HFpEF, the problem is not with the contraction of the heart, it's with the relaxation of the ventricle and the filling with blood. So we wonder if this could be traced to other problems with calcium homeostasis. So to understand that, we can go back to this diagram of the cardiomyocyte. Remember I told you how contraction is triggered. Calcium enters the cell and triggers calcium release from the SR, and that calcium binds to the myofilaments, triggering their shortening, contraction of the cell. So to relax the cell then, you have to move calcium back out of the cytosol, and this is done by SERCA. SERCA moves calcium into the SR, and the sodium calcium exchanger, which removes calcium from the cell. So these two different proteins, they return you to a low level of resting calcium. After each heartbeat, it allows the cell and the whole heart to relax. Very important. So we wondered, how is this changed in HFpEF? If you have calcium-dependent impairment of relaxation, that's what we call active stiffness. It's active, requires ATP to remove calcium, so it's an active stiffening of the ventricle. And we wondered if this happened in HFpEF. Now, you have to know, though, that in the clinic, there are all sorts of different patients, different types of patients that end up with HFpEF. So there's different disease etiologies. It's kind of a mixed bag of patients. So first we looked to model this then, we looked at different types of rats that had developed diastolic dysfunction in HFpEF. First, we looked at those that had small infarcts where they had only diastolic dysfunction. You see, when we look at the calcium transients, the normals here, JAM, looks like this, and you see that the calcium transient is very similar after infarct, but it's actually declining more quickly. So there is no problem with calcium removal here. And these two proteins that remove calcium, SERCA and NCX, they're actually working harder. They have a higher rate of calcium removal. So no problem with calcium there. Similarly, when we looked at hypertensive HFpEF, lots of HFpEF patients develop the disease after they've had hypertension. We see that the calcium transients are very similar in the rat model of hypertensive HFpEF in comparison with the controls. Again, no change here in the SERCA activity, actually a little bit more activity with the NCX still. Here's the interesting part though. Lots of diabetic HFpEF patients out there in the world. It's an increasing global burden. So many patients that have diabetes now will unfortunately eventually develop HFpEF. And when we combined hypertensive and diabetic HFpEF in this model, which is the obese cytocephalon rat in comparison with the lean rats, now when we look at the calcium transients, you see calcium goes up and comes down more slowly than in the controls. There's a slower removal of calcium, what we call an active stiffness. That's due to less expression and activity of both SERCA and NCX. So there's big differences in HFpEF depending on the type of disease etiology. If we go back to this diagram, then the problem in diabetic hypertensive HFpEF is that there's less SERCA and less NCX. There's less protein expression and less activity. So there's less recycling of calcium into the SR, less removal by the sodium calcium exchanger. So you get a slowed calcium removal and a higher resting level. And what this means is that the relaxation of the cell and the whole heart is slower. The relaxation is also less complete, both at the cell level and at the whole heart level. So there's an active stiffness. So to sum up then, this is the basic take-home message. I told you that how the system works in health, and in heart failure, there's different types of problems that occur. So in HFrEF, this condition of heart failure where the ventricle doesn't contract as well, we can link that to changes in the cardiomyocytes. The structure of the cells are basically broken. The T-tubules are really disrupted. The ryanodine receptors are broken apart. So this disruption of the cells impairs the calcium release, impairs the contraction of the cell, impairs the contraction of the whole heart. We think that an important driver for this is the high workload that's placed on the heart during this condition. What we call the wall stress, there are different ways that it's manifest. This is important because we have lots of drugs that treat HFrEF already. And what do those drugs do? They reduce workload. So you have beta blockers, angiotensin receptor antagonists, ACE inhibitors, you have ventricular unloading devices. All of these things decrease the workload. We think this is part of the reason they work. They help protect or even repair the structure of the cells. Now, HFpEF is a very different animal. Here, the structure of the cells appears to be very good. It's actually often better than normal. But still, sometimes we see problems with calcium homeostasis, and it's in the removal of calcium that's important for relaxing the cell. This is particularly prominent, we think, in diabetic F. Now, you might ask, so why do these other patients have impaired relaxation in this condition? Well, we know that it's not just the cardiomyocytes. There's also what we call passive stiffness, and that's due to changes, for example, in the extracellular matrix that stiffen the ventricle. So depending on the condition, there may be a combination of active and passive mechanisms. It's really important that we figure out these interventions because we're almost completely unable, unfortunately, to treat HFpEF patients at the moment. So I'd like to thank you all for your attention, and I'd like to thank my group members who contributed to this. Here we are at our annual skating party. It's, of course, always winter in Norway. And the people's data that I showed you here were from Dave Lipset, Mariana Ruud, Chin Chen, and Mikko Frisk. Some of you may know Ulla Seierstedt, who was my mentor for many years, now retired, but still with our group as Professor Emeritus. And I'd like to thank you all for your attention. I'm happy to take any questions.
Thank you, Bill. Very nice talk. So while we have some questions prepared, I think I'm going to ask you the first question.
Sure.
So you're saying that this impaired calcium dynamics is based mostly on the spreading apart of these ryanodine receptors.
I'd say it's in HFrEF, a combination of the disrupted structure of the teachables and the breaking apart of the renoreceptors. Yeah, right.
Yes, and have you tried modeling, taking into consideration the diffusion coefficients of calcium to see whether a mathematical model would fit these calcium delayed responses, to see whether the spreading apart fits your model?
Yep, absolutely. I've worked with mathematical modelers a lot. I don't do it myself, so I really rely on them. And super resolution imaging, they take the structures of the dyads as we see them, they put those into the model. And then they say, okay, if you activate, let's say you have a cluster of this, or a series of clusters that are nearby each other, and they see then calcium jump from one to the other, and it gives you a very slow calcium spark. And that's what we see in the experiments. Similar slowing of the calcium spark, and that promotes a desynchronization of the calcium transient. So it's a very useful technique to use modelers for.
Yeah. Right. So we have a question here from Martin Villa-Petrov.
Yes.
It's a great talk. He's asking whether there are endogenous compensatory mechanisms to ameliorate calcium handling in herbs.
Yeah, absolutely. And especially in rodents. Rodents are really good at compensating their calcium homeostasis. They seem to be better at compensating for just about anything in comparison with humans, at least. So a classic response is that mice and rats, they increase circ activity when necessary. So they have supernormal calcium removal. So when we get to a disease state, then you've really gone past the compensatory part, back to normal, and then worsened again.
Great. And the last question, because we are out of time, from Alicia Matiasi. She's asking whether it is possible to differentiate changes in the stiffness of the myofilaments, the cross bridges, from changes in the stiffness due to impaired relaxation.
Okay, yeah. So that's a good point. The cross bridges, we would guess, would go together with the calcium deficits. But I think an interesting question is actually separating the passive stiffness from the active stiffness. So you can take tissue, you can actually basically destroy the myofilaments. So you remove any active stiffness, and you can then measure the remaining stiffness, which is due in the cells to tighten, and intact tissue due also to the collagen changes in the extracellular matrix. So then you can try to calculate how much comes from collagen, how much comes from titin, how much comes from calcium. It's difficult. That's actually a good question for modelers, to try to put all those numbers together to see how does it add up to a ventricle that doesn't relax as well.
Great, thank you, Bill. I'm sorry, we have more questions, but we have to move on.
I have a question for Bill.
Yes, please.
Thank you very much, Bill, for your nice presentation.
Sure.
I wonder What do you think about pharmacological RYR2 inhibition? Maybe based on your results, where in reduced ejection fraction model, you have less dyadic or less dyads, you think it is more dangerous to treat for antiarrhythmic purposes patients with reduced ejection fraction with RYR2 inhibitors than patients with preserved ejection fraction model?
Yeah, it could be. It could be. We know that it's a difficult question. So we're starting to understand how these things could lead to alternans and such, and that if you additionally blocked RYRs in some parts of the cell, that could, depending on where you are, it could make things worse. But there are so many factors at play. It's a quite difficult question to answer.
Okay, great. Deserves
Thanks very much.
Discussion. Okay.
Sure.
Thank you, Bill. Okay, so we are moving on to our second speaker, which is Juan Goodman. Juan is an independent investigator of the National Research Council and is the director of the Laboratory of Transmisión Sináptica del Sistema Auditivo at INHEVI. Thank you very much, Juan, for joining us, and you're more than welcome to give your talk in Spanish.
Okay, can you see my screen?
Yes.
Excellent. So I was thinking in giving the talk in English, if that's okay. I can surely take questions in Spanish or whatever, but I don't know, especially for Bill, I thought it was better to give it in English. Anyways, so thanks Dario, and thanks Chris for the invitation, also Martin Milan-Petrov. I'm really honored to be here. This is my first time at Southeast, and so far it's been great. And as I said, maybe my talk is a little bit off topic for the society, but I'm sure that there are multiple similarities that I could hopefully can give us, so you can give me some feedback from the data. So I will talk about calcium in the auditory system, as the title of my talk says. And I should say that calcium is very dynamic in these cells because it underlies the encoding of sounds, which is by itself, sounds is a very changing signal in the environment. So calcium has to be very, very tightly controlled in hair cells, and I will show you some of our results in the last few years. So as I presume that not everyone in the audience is familiar with the auditory system, I thought I would start off by introducing very briefly, surely, the auditory system. And what you see here on the left is naturally a diagram of the ear. Can you see my second slide? I guess you can, right?
Yes, we can.
Okay, cool. So surely this is a diagram of the ear. This is a human ear, but it's not any different from other mammals. And it consists of an external ear, the tympanic membrane, the middle ear bones. There's the smallest bones in the body. And these bones, the middle ear is responsible for coupling airborne sounds into the changes in movement of fluid within the inner ear. Okay, the inner ear by itself can be subdivided into the vestibular labyrinths, and the vestibular system, you may know, is responsible for detection of body movements and head movements, whereas the cochlea, which is the other part, is where sounds are actually detected. Okay, so the cochlea is this snail-like structure, very peculiar. And if we made a transverse cut of the cochlea, as suggested in the image, one will see a very complex set of compartments and spaces that I don't want to go into details. But what I would like to especially point out is the organ of Corti. The organ of Corti is actually an epithelium, and it's the so-called sensory epithelium within the cochlea. And as you can see here in the image, is composed of multiple cell types. But there are two cell types that I would like to especially name, and these are inner hair cells and outer hair cells. These are both the sensory cells in the auditory, in the cochlea. And they're both responsible for transducing sounds, meaning converting sound signals into electrical potentials. And they do so by means of these specialized organelle. This is stereocilia on the apical end of the cells, they both inner and outer. So both inner and outer hair cells are responsible for this, but they have very different function as well. Inner hair cells are responsible for transmitting the information, the transfused information into the brain, and they do so by means of these synaptic connections to auditory nerve neurons. Okay, so information transfer is the main function of inner hair cells. So we ourselves, we make a lot of work on this property of hair cells, of inner hair cells, but that's not what I'm going to talk about today. Outer hair cells, on the contrary, their main function is to amplify sounds, and they do so by the capacity to change their cell length in response to membrane potential changes. Okay, they can shorten and lengthen in response to changes in membrane potential, and by that they can amplify this, particularly small sound signals. So signal amplification is the main function of outer hair cells. Okay, today I will talk about outer hair cells and calcium, as I mentioned before. Okay, so the main calcium source in these cells is actually the mechanism action channel. These channels are at the tip of these stereocilia. Only some of the stereocilia have channels in their tips. And when the stereocilia move back and forth, these channels open. Cations, both sodium and calcium can go in the cell, but calcium has to be very tightly controlled so the cell can mechanically transfuse correctly. And these are some of the properties here on the right that allow their hair cell to control calcium. They have very large amounts of calcium ATPase, these are proteins in the membranes of these stereocilia. They also have very high concentration of calcium buffers in the stereocilia, especially this buffer is called oncomodulin in millimolar concentration almost. And there's also a very important layer of mitochondria right below the area of the stereocilia. And the function of this mitochondria, among other things, really, is to prevent the calcium leak from the stereocilia compartment, from the apical part of the cell, down into the basolateral compartment. Okay, but there are also other calcium sources in these cells that were sort of neglected, in our opinion, and these are also synaptic connections that outer hair cells present. Okay, so these are epithelial cells, but they make synaptic connections with two different cell types, or neuron types. One of them are these neurons from the brainstem, particularly from this area. It's called medial olivary complex, and these neurons are cholinergic. Okay, so I refer to these neurons, these synapses. I will talk quite a bit about this in the following slides, and I will refer to it with MOC acronym. Okay, so these neurons are cholinergic. They release acetylcholine, and outer hair cells have this special type of nicotinic receptor assembled by these two subunits, and the receptor is highly calcium permeable. Okay, so every time they open them, calcium is let in the cell, and then calcium opens up, in turn, a potassium channel of the SK2 family, and these channels are only calcium dependent. There's no voltage dependence of these channels. Okay, so the overall effect of this synapse is to activate the potassium channel, producing synaptic inhibition in these cells. Okay, so this is a typical, for electrophysiology expert, this is a typical current clamp recording of a synaptic potential. You can see the downward deflection of the membrane potential. As I said, they produce inhibition, and the main role of the MLC innervation is to reduce the amplification that outer hair cells can produce in the ear. Okay, I shouldn't forget to mention that another important aspect of this synapse is the presence of these very important cistern that is present right next to the outer hair cell membrane, and I will show some electron microscopy image in a second. Okay, so on the one hand, there's calcium influx through the MOC synapse. On the other hand, I want to mention this other synapse with the so-called type 2 spiral ganglion neuron. Okay, this is a glutamatergic synapse. So vesicles filled with glutamate are released only after calcium channels, voltage-dependent calcium channels of the L type are open. Okay, so calcium influx now comes in the cell, in outer hair cells, through the voltage-dependent calcium channel. So these synapse outer hair cells operate sort of as a presynaptic compartment. Okay. So the questions that, as I said, so we found that the calcium effect of these synapses was sort of neglected, and we wanted to study and answer some of the questions that I'm mentioning, that I'm including here in the slide. So what's the shape of these signals? What's the dynamic when stimulated in different forms? And how are these signals actually measured? Okay, so the way we addressed this question was using mice. We took mice at these very early, very young ages. So we used mice of postnatal day 12 to 14. And we do that because after, so you should consider that the cochlea by itself is a bone. It's surrounded by a bone. And the older the mice gets, the harder it is to dissect out the cochlea and the organ of cortex particularly. But at this age, we feel comfortable, and sometimes we do it even in an older animal. So we dissect out the organ of cortex, and this is an image of what we see in the microscope. This is a top view of the organ of cortex here. So these U shapes, inverted actually U shapes that you see here are the stereocilia of the 3 rows of outer hairs. Okay, so we actually do electrophysiology in these hair cells. We use patch clamberentos, we patch in the cells, and at the same time we fill the cells through the patch pipette with a calcium indicator. In this set of experiments, we use fluo4, and we fill the cell with this indicator. So in this first set of experiments that I will show, what we did is to stimulate the MOC axons with the second electrode, extracellular electrode, and stimulate the axons to make them release acetylcholine. So the movie that we play here, I show is an image. It's a movie taken at the base of the hair cell with the dashed line indicates while we stimulate the fibers. Okay, so you will see a very brief. Actually, the movie is slowed down, obviously, but a very localized and small calcium fluorescence change actually at the base of the hair cell. Okay, so if we measure the change in fluorescence in a particular region of interest, we observe these changes in fluorescence, these transients, and that correlate very well with the synaptic currents that we measure at the same time in the cell. So many times we stimulate the cell, we stimulate the fibers, the MOC fibers, and there are synaptic failures from the terminal phase to release vesicles. And this occasion we see these traces. So these gray traces indicating there was no synaptic currents. At the same time, there's no change in fluorescence as indicated here. Okay, so this again is using actually a very brief and discrete stimulus pattern. But we know from literature and even our own work that this kind of stuff operates actually in trains. It operates repeatedly. And actually, the longer the train, the more inhibition it produces. Okay, to the hair cell. So we decided to look at the calcium transients, applying trains at 20, 40, and 80 hertz. And as I say, that these signals can fire up to over 100 hertz in physiological context. And as you can see, the calcium transients grow larger, the larger the frequency of stimulation, which we quantify here as delta F over F. And this is naturally a correlation between the change in fluorescence and the integral of these currents. Okay, so there's no saturation of the dye or anything that could prevent phase-loop measurements. So as I mentioned before, there's this system like this located very close to this synapse. And these are electron microscopy image by our friend and colleague Paul Fuchs, which shows first on the left, you see the base of the outer hair cell, the nucleus, and 2 different MOC terminals or efferent terminals that is indicated in this label there. So on the right, you can see actually a detail of the synapse, the contact zone between the efferent terminal, the MOC terminal, with the vesicles in the interior. And actually, you see this. You can see a set of 3 membranes because they belong to the plasma membrane of the outer hair cell. and the two membrane of the system, which is, again, as I said, very narrowly located close to the outer hair zone. Okay. So they living up only a few nanometers apart between the space between the two membranes. Okay, so there's the system that's located very close to the synapse. It's actually, that's why we gave the game to name a subsynaptic system. And this system, there was some information, some reports, I would say indirect, that there were circa pumps located in the system and ryanodine receptor. Unfortunately, fortunately to me, Bill already introduced sort of the function of these two proteins. So I don't want to go into the details, but what is important to know is that we want to directly measure now, looking at the calcium transit and manipulating the function of these two agents. So firstly, we use tapsicargine, which is a very well-known blocker of the circa pumps. And when we do that, we see an increase in the size of the current, the calcium transient, which is quantified here, and also a lengthening of the duration of the transient. We also evaluated ryanodine receptors, and these are transients using a high concentration of ryanodine, which is the known blocker of the receptors, dantrolene, that is another blocker, and the low concentration of ryanodine. Through the patch electrode, and these are some transients that you can see record when you step the hair cell from large as those obtained with the MOC stimulation. Because there were previous reports saying that there were few channels and the currents were actually negligible. We also measured the distance between calcium influx through the one synapse to the other within the same hair cell, and we come up with these individual numbers from. So the average about 3 microns, but there were many that were around the micron. The role of circa pumps and ryanodine receptors in these voltage-dependent calcium channel transits. And we didn't observe any change when applied tapsicargine, and neither when we use a high concentration of ryanodine to block ryanodine receptors. But we did observe a strong potentiation with the low concentration of ryanodine, indicating that these transits could be potentiated with ryanodine. Okay, so just to summarize, the summary, I went through the conclusions as I went through. I only want to measure that these two synapses, the MOC synapse to outer hair cells, and the one with type 2 spiral ganglion neurons, they can be regulated differently with circa pumps and ryanodine receptors in our hands. And I only want to finish off by thanking the person who actually did the experiment, student in the lab, and also for the postdoc, Marcelo Moule, who is now in the UK, an absolutely brilliant student, other members of the lab for comments and discussion. And I should also say, to finish up, that my laboratory is part of a larger group of people working in auditory questions in the auditory field. And we are fortunate to be able to collaborate with each other. And I should mention Carolina, Guedebey, and Mario Gomez. Some of the people we collaborate. Many of you may know Belén Egoyan. Belén is the founder of the group. It's like our angel guardian in some sense. She's a brilliant scientist. You may know a lot about her, but she's also a great person to work. And also should thank the funding sources to finish up. So thanks.
Thank you, Juan. Very nice talk. So we begin the question sessions for Juan Goodman. Our first question is from Alicia Matiasi, Juan. She says, a nice talk, and she's asking, which is the type of the ryanodine receptors at the subsarcolemmal systems?
Yeah, so there's some information from single-cell RNA-seq experiments in these cells. And there's data showing, so these data show that the 3 of them, ryanodine receptor 1, 2, and 3 are present, were expressed in outer hair cells. But that should also say that in situ hybridization assays showed only biennial receptor type 1, which, for what I understand, are the ones that are not calcium dependent, right? If I'm not wrong.
Okay. So,
Okay,
do you have a question, Luis?
Yes, congratulations for this nice data, Juan. And given that restriction of these calcium signals is very important in your cells, I wonder what's the effect of calcium buffering? For example, what happens with the pH or temperature? Does it affect this development in the early life?
Well, that I don't know. So that's a good point. I don't know what the temperature does to these signals. Yeah, it's something that, so all these experiments were done, so I should say that, right? All these experiments were done at room temperature, right? I don't know. So the short answer to that question is I don't know, but I'm sure that if you increase the buffer, actually we, as you may expect, when you patch the cell, you dilute the native calcium buffering, right? As I said, there's a high concentration of buffers that we wash out actually with the patch electrode. So we assume that we presume that all the calcium signals will be smaller, right? When in more physiological conditions. Actually, what we need is like push the system as much as we could to detect signals. And there were problems that the signals were actually very small, and we use rather low buffering conditions and another, simple tricks to amplify the signals.
What I think is that maybe with less calcium buffering, your signals are less restricted. So
right.
Maybe it leads to some developmental problems.
Oh no, yeah, sure. So for instance, if you knock out this calcium proteinaceous calcium buffer that I mentioned on commodulin, the cells just die. So there's a progressive death of the cells, but I don't know, within a month, I think the cells are, at least 50% of them are gone. Yes, that's absolutely true.
Thank you both. We need to move on. We're running late. Thank you very much, Juan.
Thanks.
Okay, our next speaker is Silvia Belmonte. It's a pleasure to have you here, Silvia. Thank you very much for joining us. Silvia is an independent investigator of the National Research Council, and she at IGEM Mendoza. She runs the Laboratory of Lipids and Acrosomal Exocytosis. And the title of her talk is Ceramic Induces a Multi-Component Intracellular Calcium Increase Triggering Human Sperm Secretion. Silvia, it's your time now. You're, of course, welcome to give your talk in Spanish or English as you prefer.
Gracias, Darío. Yo voy a hablar en castellano. Les agradezco muchísimo la invitación a SAFIS y estar aquí. Bueno, nuestro laboratorio estudia la exocitosis. La exocitosis es un proceso en el cual, por el cual las vesículas citoplásmicas se fusionan a la membrana plasmática y liberan su contenido. En algunos casos la secreción es constitutiva, o la exocitosis es constitutiva, entonces los lípidos y las proteínas se quedan en las membranas, porque son proteínas transmembranas, pero en otros casos la secreción es regulada, como es la que nosotros estudiamos, que es calcio dependiente. En esos casos, una señal indica a la vesícula que debe fusionarse con la membrana y liberar contenidos bioactivos. Esta secreción regulada ocurre en células especiales, o sea, células secretoras como las neuronas o las células endocrinas, como por ejemplo las de la hipófisis o los islotes de Langerhans, o las células cromafines. Entonces, la exocitosis regulada es esencial para la fertilización, ya que tanto el ovocito como el espermatozoide deben secretar su contenido para que ocurra una fertilización exitosa. Por lo tanto, en nuestro laboratorio nosotros estudiamos la secreción del espermatozoide humano. El espermatozoide humano es una célula muy particular, o el espermatozoide en general, es muy polarizada, es una célula terminal que tiene una cabeza y una cola. La cabeza prácticamente está ocupada por el núcleo, que es ADN muy compactado, el citoplasma es muy escaso y está cubierta en un 50% por un gránulo gigante que es como un capuchón, que a modo de capuchón recubre el núcleo. Ese gránulo se denomina acrosoma, es un reservorio de calcio y es el gránulo que nosotros vamos a estudiar como vesícula secretoria. Muy bien. Si miramos una micrografía electrónica en un corte longitudinal de la cabeza del espermatozoide, vemos entonces este núcleo con el ADN altamente compactado. Esta cinta que rodea al núcleo, que cubre al núcleo, es el gránulo acrosomal, es electrondenso, tiene una membrana acrosomal externa que da hacia la membrana plasmática y una membrana acrosomal interna que da hacia la membrana nuclear. El citoplasma es escaso. Muy bien. Para que ocurra esta exocitosis acrosomal, las células o los espermatozoides deben estar capacitados. Eso quiere decir que han sufrido un proceso dentro del tracto genital femenino donde existen cambios bioquímicos, biofísicos, es un proceso fisiológico que lleva a que la célula tenga una motilidad hiperactivada y además a que la célula pueda responder a un estímulo como la progesterona, que es un estímulo fisiológico que induce la exocitosis de este gránulo. Este proceso de capacitación ocurre en el tracto genital femenino, pero lo podemos simular in vitro y preparar a la célula para que responda a los estímulos exocíticos. Lo primero que ocurre cuando el estímulo exocítico impacta sobre la célula es que el gránulo acrosomal se hincha. Este hinchamiento lleva a que la membrana acrosomal externa sufra ondulaciones, esas ondulaciones intentan tocar la membrana plasmática y posteriormente en esos puntos donde tocan se producen la fusión de membranas y se liberan vesículas híbridas y por lo tanto se pierde la membrana plasmática, la membrana acrosomal externa y queda como membrana limitante de la célula esta membrana acrosomal interna. Por supuesto el segmento ecuatorial queda siempre invariable. Si lo miramos al microscopio electrónico para ver cómo es en realidad, esto es un hinchamiento del acrosoma. Se observa que pierde la electrondensidad. Estas invaginaciones y evaginaciones de la membrana se ven perfectamente. Estas evaginaciones intentan tocar la membrana plasmática hasta que hay aposición de membranas y se mezclan las bicapas lipídicas y entonces se produce la liberación de estas vesículas que son mitad membrana acrosomal externa y mitad membrana plasmática. Por eso se llaman vesículas híbridas. Acá se ve la célula vesiculizada y acá se ve tiene la membrana acrosomal interna como membrana limitante y el sector o el segmento ecuatorial queda invariable y está listo para la fertilización del ovocito. Muy bien, ¿cómo vamos a medir nosotros en los ensayos de exocitosis si el acrosoma está presente o no? Normalmente utilizamos una lectina de Pisum sativum que se une a los residuos de manosa de las glicoproteínas presentes en la matriz acrosomal y que está acoplada a algún fluoróforo. Y entonces, cuando tiñe el acrosoma podemos verlo. Hay tipos de tinciones directas o indirectas, pero lo que importa es que siempre nuestro readout es si el acrosoma está o no está y se puede contar en un microscopio de fluorescencia o mejor por citometría de flujo. Eso es lo que voy a graficar normalmente. Hace muchos años nosotros empezamos a estudiar los esfingolípidos. En realidad, los esfingolípidos son moléculas o componentes estructurales de la membrana plasmática y el estudio de los mismos se vio retrasado justamente por pensar que la única función era estructural. Y sin embargo, los lípidos son moléculas bioactivas muy importantes. Nosotros estudiamos una de las moléculas bioactivas, que es la esfingosina 1-fosfato. Esta se llama así, esta molécula bioactiva, porque en muy pequeñas concentraciones, cuando cambia en muy pequeña concentración, produce un efecto biológico muy importante. En nuestro caso, encontramos que esta efingosina 1-fosfato impactaba sobre un receptor acoplado a proteína G e inducía un mecanismo de transducción de señales que llevaba a un aumento de calcio y a la fusión de las membranas finalmente. También encontramos que estaba presente la efingosina quinasa, que es la enzima que estimula la síntesis de esta efingosina 1-fosfato. Fue un mecanismo muy interesante y realmente muy curioso para nosotros y nos despertó un montón de dudas. Por empezar, el metabolismo de los esfingolípidos es complejo, y bueno, quisimos saber si estaba la efingosina quinasa en una célula terminal, como la espermatozoide, si había otras enzimas del metabolismo. Pero además de eso, un lípido que nos interesó mucho fue la ceramida, ya que es el nudo del metabolismo de los esfingolípidos, y puede ser sintetizada de nuevo, y así también se puede interconvertir en cualquiera de los otros metabolitos esfingolipídicos. Así que empezamos a estudiar las enzimas que estaban presentes en espermatozoides, y encontramos en espermatozoides humanos estaba presente la esfingomielinasa neutra, la ceramida sintasa. También dijimos, ¿qué pasa si desafiamos la célula con una ceramida permeable? ¿Qué ocurre en concentraciones crecientes? ¿Qué ocurría? ¿Y qué pasó? Bueno, la célula sufrió exocitosis en una forma tan eficiente como la inducía progesterona. Para que la célula respondiera a ceramida, tenía que estar capacitada. Lo mismo que para la respuesta a progesterona, y esto nos indicaba que era un proceso más bien fisiológico. Ahora bien, en este momento tuvimos un pensamiento muy minimalista, digamos, o sea, dijimos, bueno, si aumentamos la ceramida en este pathway metabólico, seguramente el equilibrio se va a desplazar hacia efingosina 1-fosfato y vamos a ver el mismo efecto que encontramos con efingosina 1-fosfato, o sea, nos imaginamos que ceramida era capaz de inducir la exocitosis vía efingosina 1-fosfato. Para comprobar esta hipótesis, inhibimos la enzima efingosina quinasa capaz de sintetizar la efingosina 1-fosfato y vimos qué pasaba cuando desafiábamos las células con ceramida. ¿Qué encontramos? Que a pesar de tener el inhibidor de efingosina quinasa, la ceramida seguía induciendo la exocitosis acrosomal, y esto indicaba que la ceramida actuaba de manera independiente de la efingosina 1-fosfato. Cuando uno trabaja con lípidos tiene que tener mucho cuidado, porque al agregar los lípidos, las membranas pueden romperse. Los lípidos se introducen en las membranas y pueden generar un efecto que no sea el que nosotros estábamos midiendo. Entonces fuimos al microscopio electrónico a ver si realmente las células tenían el mismo cambio ultraestructural cuando las tratábamos con ceramida o cuando las tratábamos cuando le poníamos progesterona o un ionóforo de calcio. Y vimos que las células se hinchaban en el mismo porcentaje, un 40%, y también se inducía la reacción acrosomal típica como la que les mostré en microscopía electrónica. Contamos las células y vimos que era similar a otros procesos. Ahora bien, como nosotros hace tiempo venimos estudiando la exocitosis, sabemos que esta depende de un aumento de calcio intracelular en el cual están involucrados numerosos canales de calcio en la membrana, pero también hay salida de calcio de los reservorios, pero además de que aumenta el calcio, deben prepararse las proteínas SNERS para la fusión y la mezcla de los lípidos. Entonces, el ensamble de las SNERS es un proceso sumamente importante. Para ver si la ceramida lo inducía, antes de tratar las células con ceramida, inhibimos una fosfatasa encargada de fosforilar un factor que es el NSF para desensamblar las SNERS y hacerlas competentes a la fusión, y también inhibimos o cortamos, clivamos la VAMP, una de las SNERS, con toxina tetánica. ¿Qué pasó? Utilizamos un inhibidor de la fosfatasa PTP1B fotosensible. Al ponerle el inhibidor, la progesterona no fue capaz de disparar la exocitosis, pero cuando iluminamos con luz ultravioleta se perdió la inhibición del inhibidor de la fosfatasa y pudo revertirse la exocitosis. Lo mismo ocurrió con la ceramida. O sea que la ceramida necesita esta fosfatasa para producir estos SNERS competentes para la fusión. Ahora bien, también cortamos entonces VAM o sinaptobrevina con toxina tetánica y vimos que se inhibió en forma significativa la exocitosis inducida por ceramida. Así que tanto VAM como la PTP1B son necesarias para que ceramida induzca la exocitosis. Todos estos estos resultados nos llevan a decir que la ceramida está induciendo un mecanismo fisiológico de exocitosis. Ahora bien, otra enzima que encontramos del metabolismo de los esfingolípidos es la ceramidasa neutra. Esa ceramidasa nos interesó saber si estaba activa. Y ya les cuento por qué. Entonces, lo que hicimos fue utilizar una ceramida que tenía un ácido graso de 12 átomos de carbono, un ácido láurico, pero acoplado a un fluoróforo. Cuando la ceramidasa actúa sobre esta molécula, se libera el ácido graso, que cuando lo separamos, hay una fase detergente, una fase acuosa, el ácido graso queda en fase acuosa, corremos una TLC y separamos el ácido graso marcado, que lo podemos ver. ¿Qué vimos? Que aparecía el ácido láurico, o sea que había en el control, o sea que hay una actividad basal de ceramidasa, que era estimulada por la presencia de calcio, si usábamos un ionóforo, y además lo más importante es que al colocar un inhibidor de ceramidasas, este lograba inhibir la actividad de la enzima, y eso era importante porque queríamos probar si el aumento de las ceramidasas endógenas inducía que el agregado de la ceramida C6 que estábamos usando. ¿Por qué queríamos saber esto? Porque las ceramidas en el espermatozoide tienen cadenas de ácidos grasos, tienen la cadena muy larga, estos ácidos grasos, y son poliinsaturados. Entonces nos imaginamos que quizás el efecto de la ceramida permeable que estábamos agregando no era igual al de las ceramidas endógenas. Sin embargo, parece que sí, porque al colocar el inhibidor de ceramidasa y aumentar la ceramida endógena, se producía una exocitosis de forma... Muy bien, vamos a analizar entonces ahora la parte de calcio. Ya analizamos las proteínas que llevaban a la fusión, y ahora vamos a ver qué pasa con el calcio. La señalización de calcio durante la exocitosis y durante todos los procesos que sufre el espermatozoide es muy compleja. Voy a simplificarla mucho, diciendo que la vía canónica es que se considera que hay un aumento de calcio debido a canales voltaje dependientes, que produce... finalmente lleva a la producción de P3, esa molécula induce la salida de calcio de algunos de los reservorios de calcio, como el acrosoma o la envoltura nuclear redundante. La salida de calcio o el vaciamiento de los reservorios induce la apertura de canales operados por reservorios, y así hay un aumento sostenido del calcio que lleva a la exocitosis. Sin embargo, hace tiempo ya se conoció que había nuevos canales presentes en el espermatozoide, solamente en el espermatozoide, que se denominaron CASPER. Son estimulados por progesterona, también por el aumento de pH y el potencial de membrana también. Entonces estos canales empezaron a jugar en distintos mecanismos. Nosotros entonces decidimos estudiar qué pasaba si colocábamos ceramida en la célula, qué pasaba con el calcio. Para medir eso, cargamos las células con un fluoróforo de calcio, permeable a membranas, el fluo 3AM, y medimos el calcio en experimentos de célula única y también en población celular. En célula única se ve así se ve la célula cargada. Cuando se agrega la ceramida se comienzan a ver aumentos de calcio, algunos que van desde la cola hacia la cabeza y otros de la cabeza hacia la cola. Si lo medimos en población y tomando el medio de cultivo que nosotros utilizamos para estas células, que es el HTF, nos aseguramos que ese HTF tenga una concentración de calcio final de 2 milimolar, cloruro de calcio, 2 milimolar, y usamos siempre como control la progesterona. La progesterona induce un aumento de calcio en la población de células, eso ya era sabido, y vimos que la ceramida también inducía un aumento de calcio intracelular, y también lo hacía el inhibidor de las ceramidasas. Por lo tanto, la ceramida induce un aumento de la concentración de calcio intracelular. Dijimos, ¿qué pasa si en vez de dejar esa concentración de calcio afuera, quelamos el calcio con EGTA, disminuimos la concentración de calcio a menos de 100 nanomolar y estimulamos la célula? Si la estimulamos con progesterona, vimos que el calcio no aumentaba. Si la estimulábamos con ceramida o le colocábamos el inhibidor de ceramidasas, aumentando las ceramidas endógenas, se producía un aumento de calcio. Esto nos indicaba que la ceramida estaba induciendo un aumento de calcio intracelular debido a movilización de calcio de los reservorios. ¿Ese aumento de calcio era suficiente para inducir la exocitosis del acrosoma? No, no lo era. Podía aumentar el calcio intracelular porque salía el calcio de los reservorios, pero necesita calcio extracelular para inducir la exocitosis. Muy bien, entonces decidimos utilizar distintos antagonistas de los canales de calcio o inhibidores de distintos canales de calcio en población celular. Entonces, este trazo, acá voy a mostrar un ejemplo de uno de las figuras. Si agregamos ceramida se aumenta el calcio intracelular, pero si colocábamos algún inhibidor, a veces inhibía y a veces no el aumento de calcio intracelular. Entonces, ¿qué inhibidores usamos? Usamos inhibidores de los canales voltaje dependiente, vimos que la ceramida inducía igual un aumento de calcio intracelular. Tampoco los inhibidores de los canales IP3, como 2-APB y cetocongina, eran capaces de inhibir el aumento de calcio intracelular inducido por ceramida. Si usábamos el rojo rutenio, que era un inhibidor de los canales de rianodina, veíamos un efecto, disminuía el calcio intracelular o no se aumentaba igual. Lo mismo si usábamos inhibidores de los OX y lo mismo si usábamos inhibidores de Caspr. Pero bueno, estos canales se supone estarían involucrados en el aumento de calcio inducido por ceramida en estos primeros 100 segundos que nosotros medimos. Ahora, ¿qué pasaba si queríamos ver si inhibían o no la exocitosis inducida por ceramida? Los canales voltaje dependiente evidentemente no intervienen, sin embargo, todos los otros canales son necesarios para inducir la exocitosis acrosomal. Por lo tanto, planteamos un modelo un poco simplificado de todo lo que pensamos, y es que la ceramida pareciera inducir la entrada de calcio por los canales Caspr y también la salida de calcio de la envoltura nuclear redundante por los canales de rianodina. Ese vaciamiento del reservorio una entrada de calcio por los canales operados por reservorio, y ese aumento de calcio ya llevaría a la producción de IP3 y salida del calcio acrosomal, y también todos estos procesos activarían el ensamble de las NERs que permitirían la fusión de membranas y así la liberación de las vesículas híbridas y del contenido acrosomal, dejando la célula lista para la fertilización. Entonces planteamos que la ceramida en realidad actúa en estadios tempranos, produciendo este aumento de calcio probablemente desde la región del cuello o de la cola hacia la cabeza. Muy bien, estas son las personas que trabajaron en esta ceramida, particularmente la doctora Cintia Baquer, es la mitad de su tesis doctoral, también la doctora Swayman, el doctor De Blas y el doctor Pavarotti. Agradezco el financiamiento de estas instituciones que confían en nuestro trabajo, Agencia CONICET y la universidad, y bueno, les agradezco un montón por su atención y por la invitación. Muchísimas gracias.
Gracias, Silvia, un gusto escucharte como siempre, muy linda charla. Vamos a las preguntas. Pregunta Alicia Matiasi, dice que es muy interesante la charla, pregunta si no podría haber un mecanismo de liberación de calcio inducido por el propio calcio, como en el corazón.
Sí, podría ser, o sea, nosotros, nos quedan un montón de dudas con respecto a qué efecto en realidad tiene la ceramida. Probablemente hay un ingreso de calcio, probablemente este mecanismo se potencie con el de progesterona, y pensamos que a lo mejor la entrada de calcio por los Caspr también estimule la salida de calcio de los de rianodina, pero en realidad también el mecanismo por el cual la ceramida activa los canales no lo tenemos claro, la bibliografía tampoco es clara en cuanto al tema. Hay un montón de canales que se supone que activa o que no activa ceramida o que los inhibe, entonces es muy controversial el tema. Sí, yo creo que podría ser, pero bueno, no tengo una respuesta digamos, firmes a esto. Son todas hipótesis.
Gracias, Silvia. Tengo un par de preguntas yo, por favor.
Sí,
Una,
dale.
si probaron si hay un sinergismo, así como vos recién dejaste entrever, si vieron un sinergismo entre ceramida y progesterona en la reacción acrosómica, esa es una. Y la segunda es, teniendo en cuenta que la progesterona genera un primer aumento de calcio, un flujo de calcio, que eso no te lo está generando la ceramida, que iría directamente al vaciamiento de reservorio intracelular, si es que la ceramida podría ser decapacitado, pero que no esté hiperpolarizado, clampeado, digamos, en un estado depolarizado, porque estarías bypaseando ese primer requerimiento.
Bueno, a ver, primero vamos a ver si te respondo la primera. Nosotros pensamos que probablemente tanto progesterona como la ceramida estén actuando en una vía que converge al menos en los mismos puntos. Y eso es interesante porque no ocurre con otros inductores, como por ejemplo la efingosina 1-fosfato, tiene una vía diferente. La progesterona no requiere efingosina 1-fosfato, pero sí parece requerir ceramida, y no les he contado el resto de la historia, pero ceramida 1-fosfato también. Entonces, la progesterona estaría activando los CATSPER por el mecanismo que ya sabemos, o sea, liberando la inhibición del araquidinoilglicerol, pero la ceramida sola también lo está haciendo. Pero cuando las ponemos juntas hay un aumento de la respuesta en la exocitosis. No sabemos qué quiere decir, hay un aumento de la respuesta a la exocitosis acrosomal cuando ponemos progesterona y C6 juntas. También pensamos que la progesterona, al estar liberando el ácido araquidónico cuando hidroliza el araquidonoilglicerol, ese ácido araquidónico es un activador súper potente de la efingomielinasa. Entonces pensamos que la producción de ceramidas endógenas potencia el efecto de progesterona, porque yo no me imagino que hay ceramida dando vuelta por ahí para estimular, sino que se produce por la célula. Entonces, esa sería una respuesta. Y la otra, no sé si respondo a lo que vos me preguntaste.
Sí, de alguna manera se responde. Me gustaría ver experimentalmente qué pasa si se capacita igual un espermatozoide, pero se le clampea el potencial, cosa de mantenerlo depolarizado, si aún así responde a la ceramida, porque estarías bypaseando
Sí,
el primer...
Sí, eso no lo sé, pero lo único que hemos hecho es sacarle el calcio de afuera.
Sí.
Entonces, si yo digo que los canales de rianodinas están activando por CATSPER, porque entró calcio, y le saco el calcio de afuera y lo mismo se activan, ¿me
Sí,
explico?
eso suponiendo que el primer y segundo influjo sean por el mismo canal, por CATSPER.
Claro, suponiendo, entonces probablemente, bueno, el CATSPER entre en otros cationes, porque es un canal catiónico. Pero bueno, no sé, la verdad, no me imagino lo que podría pasar, pero bueno,
Gracias,
experimentalmente podríamos probarlo.
súper interesante. Luis, no sé si tenés alguna pregunta.
Sí, bueno, Silvia, muy linda tu charla. ¿Me escuchan? Sí, ¿no?
Sí, gracias, Luis.
Quería consultar respecto de la inhibición de los receptores de renodina, que vos usaste Ruthenium Red, si te has planteado tal vez la posibilidad de usar otros inhibidores, porque pienso que hay muchos fármacos que se plantean como potenciales antiarrítmicos, y en base a tu charla digo, ¿podrían producir infertilidad estos, o sea, tal vez valdría la pena probarlos, ¿no?
Sí, tendríamos que probar, podríamos probar un montón, si tuviéramos un montón, hemos probado lo que teníamos y algún otro experimento que tenemos por ahí, pero no he probado un montón de inhibidores, pero sí, es súper interesante el tema, ya que, bueno, no está descrito esto en la literatura, pero porque los canales de rianodina tampoco han sido muy estudiados en la exocitosis. En realidad recién ahora está tomando como más auge el CATSPER, el receptor de rianodina. En general siempre se consideraron los canales de IP3 y como el reservorio más importante de calcio es el acrosoma, que tiene mucho calcio, es una vesícula muy grande, y como es un pequeño reservorio, casi no se le ha dado importancia. Pero cada vez hay más canales más cosas que van apareciendo nuevas y que en realidad antes no se han tenido en cuenta y que ahora probablemente uno tenga que pensarlas de otra manera.
Bueno, si te interesa, en nuestro grupo tenemos algunos inhibidores, así que tal vez podríamos colaborar si hace falta, o nada, prestarle algún inhibidor para ver qué pasa.
Me encantaría, estoy abierta a un montón de propuestas y de cosas que uno puede hacer. Siempre uno termina un trabajo, pero te quedan un montón de preguntas por responder, un montón de cosas nuevas que nacen y que se generan más dudas. Y bueno, siempre, nunca se termina. Por suerte, nunca se termina y uno siempre está entusiasmado para hacer más cosas. Y eso es lo lindo de esto, que cada trabajo genere preguntas.
Bueno, muchas
Genial,
gracias. Gracias, Luis.
Gracias a ustedes.
Nos movemos entonces hacia nuestro cuarto speaker, es la doctora Silvina Ponce Dawson. La doctora Ponce Dawson es investigadora superior del FIBA. Quiero remarcar que es la presidenta designada a cargo de la Unión Internacional de Física Pura y Aplicada, y es la primera vez que alguien de Latinoamérica ocupa el cargo y la segunda vez que una mujer preside esa unión internacional. El título de la charla de la doctora Ponce Dawson es Cuantificación e impacto fisiológico del flujo de calcio intracelular. Luis, te voy a ceder a vos la manija porque tenés la charla de la doctora, creo.
Bueno, no sé si la doctora Silvina quiere que directamente hagamos el intento con el PDF que me pasó.
No sé.
¿Quiere intentar compartir su pantalla? Creo que tenemos tiempo para intentar compartir su pantalla.
Bueno, intento compartir y si no funciona, vamos con tu PDF.
Ok.
¿Se ve?
Sí.
Ok. Switcheo al inglés mejor, ¿no? Porque hay todavía o no hace falta, decime.
Como te guste,
Okay.
como prefieras.
Okay. So the title was slightly different, I think. This is work that I've done with some former students of mine, Estefanía Piegari and Cecilia Villarroel, and these are Estefanía, Lucia, and Cecilia and Guillermo, because it was based on previous work we had done with Lucia and Guillermo. So let me know if it's okay. If not, we switch to the other. Is it okay?
No,
So far, so
Okay. Okay, so this is a motivation that you don't need, which is calcium is involved in so many things. So my background is from physics, and I've come to this subject from the modeling point of view. So maybe my approach is very different from what you've been looking at. Anyway, but the main question we tried to answer was what makes it universal and specific. And basically, the way we think of this is that you have different spatiotemporal patterns of the intracellular cytosolic distribution, calcium distribution, that can induce different end responses. And that is key for the universality of the signals. And so great part of the work we've been doing was on those aspects. And in particular, we looked at signals that involved calcium release from the endoplasmic reticulum through IP3 receptors, like in the case of the fertilization wave that I have there, that I'm going to talk a little bit more afterwards. So this is an example of some type of signal in which the different spatiotemporal distributions of the calcium, intracellular calcium concentration yields different end responses. This is a synaptic modification through some sort of protocol that you do to one synapse and then induce, this is the one that you challenge with a particular perturbation, and then you look at whether some other synapses change their strength. The way that you change the strength of the synapse at the heterosynaptic terminal depends on whether there is a calcium wave that is able to propagate from one spine to the other. So if it remains localized, you do something different to this other synapse than if there is this calcium wave. So, as I told you, we are looking at calcium signals that involve calcium release through IP3 receptors, and we are interested in looking when waves are able to propagate, and not only that, but also the nature of the waves, whether they are saltatory or continuous. And we look at the model system, basically Xenopus laevis, oocytes, immature, and eggs. And because the characteristics of the intracellular waves in these two systems, it changes with maturation. And so what we tried to do was to combine modeling and experiments to try to understand what was the key ingredient that produced this change in the type of propagation. And we call it hardware rewiring, but we can. So let me talk a little bit about the IP3 receptor. I don't know if this is necessary. So this is a ligand-gated channel that is located on the membrane of the endoplasmic reticulum, and it is one of the main targets for the initiation of intracellular calcium signals. It is sensitive to IP3, so the open probability increases with IP3, and it also depends on the calcium concentration, but in a biphasic way, in the way that it's increasing the probability for low calcium or intermediate calcium concentrations, and then the open probability decreases for larger ones. So this is a very schematic model with two sites for calcium, one that activates the channel, induces its opening, and another one that inhibits it, and then a site for IP3. But this ability of a calcium, cytoplasmic calcium, to induce the opening of the channel when the concentration is not too high, gives rise to calcium-induced calcium release, as in the ryanodine receptor. So, and in many cell types, these channels are organized in clusters, and so this non-homogeneous distribution of the receptors, together with CICR, gives rise to a variety of signals, because if you have very few channels that have IP3 bound, then if one of them becomes open and releases calcium, there are not too many nearby channels with IP3 bound, so you cannot propagate the wave. But if you have many channels with IP3 bound, then you are able to propagate the wave through CICR. And so because of this non-uniform distribution, is that eventually in some cases you can have what is called propagation failure. So this is an image, this is from Ian Parker's group, this kind of fireworks there. This is a two-dimensional image in an oocyte, and those sort of fireworks are localized sites of calcium release. They are called calcium puffs, like the sparks in ryanodine receptors. Same thing, and suddenly you will see a wave that is able to propagate. It comes now here. Okay, great. So at some point many years ago, we tried to understand the key ingredients of this propagation failure and these different ways of propagation, with a very simple model that had the sites of release distributed along a line that was equally spaced. I mean, the distance among them was the same. And so depending on how this distance compared with the diffusion coefficient at the time that the channels remain open, we could have two types of propagation, what we call saltatory. This would be like snapshots of the wave at various times, or continuous. These are two snapshots, and you see that the wave propagates almost without deformation. And that depends on whether the sites of release are distinguishable or not, basically. So, and the numbers we used for that model were taken from the waves in immature eggs, in immature oocytes, and in eggs. So this is again the calcium wave. And so what are the key ingredients, we asked, that change when you go from the immature oocyte to the egg? And we thought really that the thing was the redistribution of the IP3 receptors, because this is it happens, and these are experiments that were published by some other group that shows a redistribution of the, of the IP3 receptors in the oocyte and in the egg, with these patches of high density and more continuous of the receptors. But there are other changes also with maturation, like some pumps that extrude calcium out of the cell. They are also invaginated, and so they don't work. But we were looking mostly at the spatial distribution. We thought that was the reason. So we repeated some experiments like the ones that in this paper had been done, which is you can mature the oocytes with progesterone and perform experiments in oocytes and eggs. And this is the way we do the experiments is using caged IP3 that we photorelease with a UV flash. And we applied 2 UV pulses, so we released IP3 twice. And what you see, well, is a lot of fluctuation, but you see that more patches that turn on in the oocyte than in the egg, where you see all these. The egg is more continuous all the time. There is the flickering, but there is more continuous. And so that's what we were looking at. And these are some snapshots of the experiments done in the oocyte with the very localized release of calcium. And then this is some time in between the 2 UV flashes that we applied, and you see that there are these puffs that suddenly arise, and we didn't see that in the egg. So these oocytes matured with progesterone. And so then we looked at let me go back a little bit. We picked 2 ROIs both in the egg and in the oocyte that were nearby, one to the other, and looked at whether there were differences between both of them. And we've seen that, in particular, this is from the oocyte, this is from the egg. We saw more variability in the oocyte than in the egg, and we saw that the way that the calcium decayed after the UV pulses were turned off was very different. And so we tried to fit those decays, and we observed a fast decay in the case of the oocyte. We could fit, this is the fast one. We could fit with a mono-exponential fit, but the best fit for the oocyte was a bi-exponential, and there was a very fast component. And in the case of the eggs, the time scales were much larger, so much more slowly the way it decayed, and also the best fit was linear. And this I think I'm going to skip. And so we observe larger variability in the oocyte, looking at different regions of the oocyte. Then in the time course of the fluorescence, the 2 contiguous regions, also we observe more variability in the oocyte than in the egg, and we observed a very fast component of decay in the oocyte compared to the egg. And so that fast component, we associated this faster decrease, we associated with, we think that it's related to diffusion, because if you have gradients of concentration, diffusion helps you turn them down, so dissipate those gradients. And in the egg, we didn't see that because everything was more uniform. And now in the egg, and also when the diffusion has already acted, other mechanisms of removing calcium, of lowering the local calcium concentration, and some of them is buffers and then is pumps. And that linear decay, we associate that to some sort of pumps being saturated, which is consistent with the fact that you have fewer pumps to remove calcium in the egg than in the oocyte. Okay, so low. What we then analyzed was with a very simple model in which we tried to mimic very simply what's going on in the experiment. We assumed that there were like clusters of calcium release that could induce the opening. One cluster opening could induce the opening of nearby clusters, and then we looked at the size of the cascades. Basically, we looked at how many clusters were involved in this coupled calcium release. And we did this changing the distance between the clusters and changing also the rate at which calcium was removed in the model. And well, these are the numbers. I'm not going to go into a lot of detail, but basically what I want to mention is we did two types of simulations with a certain distance, so large distance, that we were trying to mimic the oocyte, and then other simulations with shorter distances between clusters that we thought were related to the egg, that everything seemed more uniform. And then we tried different rates of calcium removal. This is the rate of calcium removal, larger or smaller. And we computed the distribution of clusters that participated in the event. And basically what we saw was that the main factor that changed the distributions rather than the distance between the clusters was the rate at which calcium was being removed. And these are some snapshots of the simulations of the calcium distributions that we saw. This corresponds to a large intercluster distance. This corresponds to a smaller intercluster distance, but with a large rate of calcium removal. And we still see some inhomogeneities here. And this is with a more slowly removal of calcium, and we see everything is slightly more uniform. So, well, this is like a little bit of everything all together. And so going to the summary, well, calcium is ubiquitous, and in many cases involves calcium release through IP3 receptors, and you have CICR in this case. And in many cell types, you have these IP3 receptors that are organized in clusters. And so the signals can propagate throughout the cell or not, depending on whether they can, I mean, you have these patches of receptors, whether enough calcium arrives at these other clusters to induce the release from those channels. And so this geometry we call, that is how it is distributed in space, is important for whether the wave is going to propagate or not. And that distribution changes with maturation, at least in the case of the cell that goes from the oocyte to the egg. And so the thing is that our approach was assuming that this interspatial distance of the distance that separates two contiguous clusters was the key ingredient. But through the experiments and the model, what we saw was that the most important aspect was calcium removal, that if calcium is not removed fast enough so that you keep some concentration of calcium that couples the clusters, then you can couple more efficiently because you already have some calcium that helps you propagate the wave and have this calcium-induced calcium release. So anyway, I think that an interesting aspect is that IP3 receptors give the cells this flexibility to change the nature of so generate. So this is the talk, and I hope you could understand me because this was I was very worried with my connection, horrible.
Oh, thank you, Silvina. It worked perfectly. Okay, so I think we have time for at least one question, if there is.
I might ask something. Selena,
Yes.
This was very nice work. It's fascinating to see how some of the same issues that you're thinking about and looking at are the same things we think about, for example, in cardiomyocytes. It's the exact same issues. And I wanted to ask you, when you were comparing these different situations, as I understand it, the IP3 receptor is regulated also by luminal calcium, correct? And do you think that there could be any differences there that might explain the differences between the oocyte setting?
What difference there are in luminal calcium, but there have been some controversies about what is the effect of luminal calcium on IP3 receptors, if it's something like, if it really affects or not. And we've also worked on trying to look at luminal calcium in the oocyte, in the mature oocyte. And basically we found that it was so much calcium that it was not on the signals on the side, basically. Yeah.
Okay.
So
Thanks.
because I think in the oocyte, let me tell you something that we came up with compared with the SR that I know, because in muscles, you have a relation between area and volume, larger, the area to volume relation. That's what we came up with, the idea that maybe in the oocyte you have a larger volume. So it's like you have more availability to, and you don't exhaust that much your source. But yeah, that was what we came up with, the idea. I guess we will have to do more things to check it. Yeah, that area and volume, the relationship.
Thank you, William. Last question, Silvina. Alicia Mattiassi says, great talk, and she asks whether you have considered the wave propagation model, fire release, uptake, fire.
Uptake. Well,
yes.
These simulations that we did, we are assuming being removed, and the model is simpler in the sense that the whole propagation is like, we are only considering how many things can be coupled. It's not that we are following the propagation in time. It's like an estimate of how many clusters would be able to release, and it's like happens immediately. So it's even simpler than the fire diffuse fire. And so we have not taken, and when we did the fire diffuse fire model, we were looking at the front propagation, so we were not so worried about the uptake. Basically, we thought that would occur on a much more, on a slower time scale.
Great, thank you very much. We're running late, so we have to end up the symposium here. I want to thank all the speakers again for this excellent symposium and great talks. And let's keep on with the program of the Congress. See you all. Bye-bye.
See you. Thank you very much.